Hi guys, I want to introduce a new topic to you. It's a very important topic as far as biochemistry is concerned, and this is nucleic acids. Now, as far as nucleic acids are concerned, there are some objectives that we expect that by the end of the day, we should be able to attain those objectives. Now, the first objective is that you should be able to define what nucleic acids are. Besides that, you should be able to state some basic functions of nucleic acid in acids in living organisms. You're supposed to explain what nucleosides are and distinguish them from nucleotides, and also be able to describe the structure of DNA and RNA, and by extension, you should be able to distinguish between DNA and RNA. The other objective for this topic is to be able to describe the processes involved in synthesis of DNA and RNA. Second last, you're supposed to be able to explain DNA sequencing and a bit of recombinant gene technology. The very last thing that we expect you to be able to do at the end of this lesson is to explain what the genetic code is and the implications of the genetic code. So those are the few objectives that we aim to attain by the end of this lesson. Now, if there be any other that we think that you need to grasp along the line, of course, I will bring it out. Now, let's get started. The first thing, we need to have some basic understanding of what nucleic acids are. Now, we talk about nucleic acids. We are talking about the key components of the genetic or hereditary material. That part of the cell that transfers characteristics of a species from one generation to another. The very basic principle of that is the nucleic acid, the basic unit, the basic component of genetic material. Now, nucleic acids are usually present in the in the cell as nucleoproteins. By nucleoproteins here I mean some protein material which is conjugated with nucleic acids as the prosthetic group. Now, in eukaryotic cells, nucleic acids are normally found in the nucleus. This is something that I'm sure you are all familiar with. Basically, we have got two types of nucleic acids. The ribonucleic acid, or basically RNA, and the deoxyribonucleic acid, what you commonly call DNA. Now, chemically, nucleic acids are large molecules, or we call them polymers of repeating units. The repeating units are called monomers, and these monomers are usually nucleotides. In essence, we are saying that a nucleotide is the basic unit of a nucleic acid. So you talk about a nucleic acid molecule. You're talking about a large molecule consisting of several nucleotides linked end to end. That's why we are saying that nucleic acids are polymers of repeating units, which are in this case called monomers. For that case, you can also refer to nucleic acids are polynucleotides. Now, this is the basic structure of what a nucleotide looks like. A nucleotide consists of three basic components. The first component is a phosphate ion or a phosphate molecule. The second component is a deoxyribose sugar. A deoxyribose sugar. Later we'll explain what a deoxyribose sugar is. And thirdly, all nucleotides have got a nitrogenous base. That's a nitrogen containing molecule, and there are basically four types of nitrogen containing molecules, which are named as adenine, thymine, cytosine, and guanine. Okay. Now, what are the components of nucleic acids? We have seen that there are basically three basic components. Now, the first component is a nitrogenous base. Okay. The other one is a pentose sugar. For example, we talked about a deoxyribose sugar. That's a pentose sugar. Then we have got a phosphate ion or phosphoric acid molecule. So these are the basic three components, and you can see the molecular structures. These are phosphate ion. Okay, this one is pentose sugar or a 5-carbon sugar if you like, and we have got a nitrogenous base. There are two types of nitrogenous bases we'll be looking at. The first one is called a purine, or the first group. It's a group of nitrogen containing compounds we call purines. The other one is a group we call pyrimidine. So these are the three components that make up any type of nucleic acid. Okay, a nitrogenous base, a pentose sugar, and a phosphoric acid. Now let's explore each one of them at a time, starting with nitrogenous bases. I've said there are different types of nitrogenous bases. The first type is what we call the purines. The second group is what we call pyrimidines. So the two types of purines are, one of them is adenine. We normally abbreviate it as A, and guanine, what we normally abbreviate as G. Now, purines are three types of purines. One of them is called cytosine, usually abbreviated as C. The other one is thymine, usually abbreviated as T, and uracil is the third purine. Therefore, there are two purines and three pyrimidines. These are the 5 types of nitrogenous bases that make up nucleic acids. So all nucleic acids, not this, all nucleic acids contain adenine, they contain guanine, and they contain cytosine, all of them. Now, thymine is only found in DNA, not in RNA, and this should be the first distinction between DNA and RNA, in that DNA, besides adenine, guanine, and cytosine, it also has thymine, whereas RNA, besides cytosine, thymine, and adenine, guanine, and cytosine, also has uracil, which is not found in DNA. Now, pyrimidines contain 2 nitrogens in a single 6-membered aromatic ring. We mean such a type of a ring, this is what we call an aromatic ring, and has got nitrogen, 2 nitrogen atoms in the ring. So it contains 2 nitrogens in a 6-membered aromatic ring. Now, purines have got 4 nitrogens in a fused string structure consisting of 6-membered ring and a 5-membered ring. Please note the difference. Pyrimidines have got only 1 ring, a single aromatic ring, and this ring has got 2 nitrogen atoms. Purines, on the other hand, has got 2 different rings which are fused together to form one whole structure. And within these 2 rings, or in between these 2 rings, we find a total of 4 nitrogen atoms. And here is the structure of typical purine. So that we have got 1 nitrogen, we have got 2 nitrogens, we have 3 nitrogens and we have 4 nitrogens within the ring structure. Now these 2 rings consist of one ring which has got 6 carbon atoms. Like if you begin here, that's carbon atom number 1, carbon atom number 2, we have got a carbon number 3, we have a carbon number 4, we have a carbon number 4, we have 5, 6. Okay, that's 6 member means in the ring structure we have got 6 atoms. So this 1, 2, 3, 4, 5. So this is 5, atom number 5, and then this is atom number 6. So this ring has got 6 atoms. This other ring has got 1 atom. Here we have got 2 atoms. Here we have 3. We have 4, and then we have 5. So this one has got 5 atoms in the structure. This other ring has got 6 atoms in the structure. Okay. So if you look at specific purines, the 2 purines are adenine and guanine. You can see each one of them has got 2 rings. The first ring with 6 atoms in the main ring structure, the other one with 5 atoms in the main structure. So this one here is adenine, this adenine, okay, and then this one here is guanine. So adenine and guanine are both purines. So these 2 are purines. Now pyrimidines, we have got 3 types of pyrimidines, and I told you earlier the difference between a pyrimidine and a purine is that a purine will have 2 rings but fused together. Okay. Pyrimidines, on the other hand, have got only 1 ring, and that ring has got 2 nitrogen atoms in the ring. Okay, remember you're not counting those which are outside the ring. Like if you look at the case of this is cytosine. Cytosine has got the main ring structure. This is what you're calling the ring. Okay, with the ring structure. This ring structure has got 1 nitrogen and has got 2 nitrogens. Okay, the same to thymine. Thymine, remember I said that thymine is only found in DNA, not in RNA. Thymine has got 1 nitrogen, it has got 2 nitrogens. Uracil here has got 1 nitrogen and 2 nitrogens. Okay. The differences between the 3 are very minimal. Specifically, you look at the position or the presence of the carbonyl groups and amino group. Cytosine has got 1 carbonyl group, carbonyl. Carbonyl, this compound, a carbon atom bonded to oxygen by a double bond. This is what we call a carbonyl. Carbonyl is like this. This is carbonyl. Carbonyl. I'm sure you know this carbonyl. Okay, that's carbonyl. So we have got 1 carbonyl group and 1 amino group in cytosine. Now look at thymine. Thymine has got a carbonyl group, and beside the carbonyl group we've got a methyl. This was called a methyl group in thymine. Now uracil has got 2 carbonyl groups. This carbonyl group number 1, this one is carbonyl group number 2. Okay, hope you got the distinction between cytosine, thymine, and uracil, right? Cytosine has got 1 carbonyl group and an amino group. Thymine has got 2 carbonyl groups, 2 carbonyl groups. Okay, there's another carbonyl group here. I forgot to mention that. 2 carbonyl groups and a methyl group. Uracil has got 2 carbonyl groups, does not have a methyl group. Okay. Now looking at the pentose sugars, that's the second component of a nucleic acid. Again, there are 2 types of pentose sugars. Pentose, I told you, means 5 carbons. So these are sugar units which have got 5 carbons in their ring structure. The ring structure, or remember the Howard projection. Okay. So the distinction between the 2 types of pentose sugars, that is ribose and deoxyribose, is that if you look at the structure of ribose, it has got so if you look at carbon 1 of ribose, this is carbon 1 of ribose, and this carbon 1, this carbon 1, then carbon 2. Now you look at carbon 2. At carbon 2, we have got a hydroxyl group, that's OH group. Now compare that one with carbon 2 of deoxyribose. You'll notice something important here, and this is the basic distinction between these two. At carbon 2 of ribose, we have got OH. At carbon 2 of deoxyribose, we have got H, meaning that deoxyribose is lacking 1 atom of oxygen. That's why we say deoxy. We say deoxy. D meaning without. Without oxygen. Without. So deoxy, without oxygen. So the difference between deoxyribose and ribose is that deoxyribose has got 1 atom of oxygen less compared to ribose sugar. Basically, deoxyribose is found in DNA and ribose sugar is found in RNA. Okay, thank you for watching this clip. We have basically looked at what nucleic acids are and the basic structure of nucleic acids. Basically, we have looked at the I can say that there are 3 basic components: a nitrogenous base, a pentose sugar, and a phosphate ion. So we have discussed at length the structure of the nitrogenous bases, and there are 2 types: purines and pyrimidines. Then I've discussed with you the structure of the pentose sugars, and there are also 2 types: ribose and deoxyribose. Ribose is found in RNA. Deoxyribose is found in DNA. Next time we'll look at the last component, which is the phosphate ion, which I'm sure you all know what a phosphate ion is. PO4, phosphate ion, written like this. It's just the normal phosphate. You must have learned about this in your high school chemistry. This is phosphate, usually with a net negative charge. So this is the 3rd component of nucleic acids. So we'll stop there today. We'll continue with our lecture in our next lesson. I wish each one of you a good time. Thank you.